Hello everyone. Welcome to today's live broadcast, Extracellular Vesicle Isolation by Flow Cytometric Sorting and Characterization by Analytical Ultracentrifugation and Dynamic Light Scatter by Dr. Carley Ross. I am Dr. Chad Schwartz, Senior Application Scientist at Beckman Coulter, and I'll be your moderator for today's event. We are delighted to bring you this educational webinar presented by LabRoots and sponsored by Beckman Coulter. Beckman Coulter is a leader in centrifugation and flow cytometry and has long been an innovator in particle characterization, lab automation, and genomics. Beckman Coulter products are used at the forefront of important areas of investigation and discovery. For more information, please visit beckmancoulter.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the icon in the lower right-hand corner of the slide window. If you have any technical problems viewing this presentation, please click on the support button at the top right of your presentation window or submit your problems through the green Q&A button. I would now like to introduce today's speaker, Dr. Carley Ross. Carley is a staff development scientist from Beckman Coulter Life Sciences Research and Development since 2008 and aided in the development of the MoFlow Astrios and Astrios EQ, as well as her current work on the analytical ultracentrifuge. She has been awarded 2 patents, one in flow cytometric sorting and the other for extracellular vesicle work on the AUC. In addition, she has been awarded the Corporate Excellence and Values for her work ethic and 2 Excellence in Innovation for her collaborative work on projects for the Astrios. Dr. Ross received her PhD from Colorado State University from the Cell and Molecular Biology program with a focus on mammalian mutation assays on a flow cytometer. She then did a postdoctoral work in biochemistry by studying the development of yeast prions with a focus on molecular and protein science. Dr. Ross's focus is to bring Beckman Coulter's technology to the cutting edge with small particle detection of extracellular vesicles on both the flow cytometry and AUC platforms. Before we get started today, I would like to ask a few poll questions. Our first question is, what is your current isolation method for extracellular vesicles? Please select any of the 5 different answers there. We'll take a few moments and then I will report the results. It looks like 36.1% actually do not study EVs. They're probably here for the flow cytometry part. 25% use differential centrifugation. 16% use density gradient centrifugation. 19% capture their exosomes and extracellular vesicles using other methods. Our next question will populate shortly. What size particles do you analyze using flow cytometry? Again, multiple answers are allowed here. Just a few more moments and we'll get the answer to this question, this survey. Most people are studying particles between 100 and 500 nanometers in size using the flow cytometer. That's pretty typical. 25% of you don't know. 20% are between 500 nanometers to 1 micron, and 11.4% would be greater than 1 micron. One final poll question, and that will populate shortly. Have you considered using analytical ultracentrifugation or dynamic light scattering to analyze the size or purity of your particles? It's a simple yes or no question. a few more moments, and it looks like 69% of you say yes, that you have considered using these two different types of technology to study your particles. Thank you everyone for your answers. I will now turn it over to Dr. Ross for her presentation.
Thank you, Chad. Today I'm going to be discussing extracellular vesicle isolation by flow cytometric sorting and characterization by analytical ultracentrifugation and dynamic light scatter. I worked in collaboration with two scientists, Dr. Thomas Ramin and Dr. Alexander Kaczynski at Beckman Coulter Life Sciences Research and Development in Fort Collins, Colorado. I will review three different topics in this presentation. Flow cytometry and extracellular vesicles. Can your system really see EVs? I will also discuss bulk and single EV isolation, and then finally focus on analyzing EVs with current and new techniques. Just as a quick review, what are EVs? As seen in this diagram, which is commonly seen in most of our presentations about extracellular vesicles, there are three main categories of EVs. Exosomes, which is over here on this side right here, microvesicles, and apoptotic bodies. So we have exosomes, we have microvesicles, and we have apoptotic bodies. Now the extracellular vesicles that we're interested in are secreted membrane vesicles, heterogeneous in size and composition. The exosomes in particular are 50 to 100 nanometers in diameter. They're released via exocytosis of multivesicular bodies. They're mostly characterized through immune cells, and they function as direct contact between surface molecules of vesicles and cells. So specifically we're interested in these, but what type of technologies could be used to visualize these EVs? Most of the people that have talked about extracellular vesicles look at them either on some sort of flow cytometer, or they look at them on some sort of nanoparticle device. Today I'm going to be focused on flow cytometry and on the analytical ultracentrifugation and dynamic light scatter. So our flow cytometer, when you look at it, we have to make sure that it has very sensitive site scatter and very sensitive fluorescence detection. On the system, this slide shows are similar. For those of us that run flow cytometers, we're very comfortable understanding what 8-peak beads are. This is an 8-peak bead with FTSE and PE. Now that's usually measured to measure MESF values, but our overwhelming question always is, do you really know how many fluorescein molecules are on your EV? Here's an example of the FTSE MESF beads from Spherotech. We can see that there are numbers located for each one of these peaks. This is the MESF, or molecules of equivalent soluble fluorochrome. We have 2,900, and at our detection threshold, only 76. Where does your exosome or extracellular vesicle fall within that range? When you are running your extracellular vesicles, it's very important that you use these calibration techniques to be able to determine your fluorescence sensitivity on your system. We can see here on this histogram that we have the 561, 579 log height versus the number of counts. We have three different populations. This particular population is our human serum extracellular vesicles. We have the we run 8 peaks with these fluorescently stained EVs simultaneously, and this shows your detection threshold. We have the unstained EVs and the stained EVs and our 8 peaks. It may actually be required for publication of any extracellular vesicle data. So therefore, this particular information is important for users to understand that it needs to be run when running your samples. Defining instrument calibration standards helps determine flow cytometry's small particle performance. There are 4 different areas I will discuss today: polystyrene beads, liposomes, purchased exosomes, and the electronic pulsar. Each of these standards can be used to determine if the flow cytometer can measure and see EVs. Running polystyrene beads for initial determination of optical sensitivity to EVs is a good start. As seen in the next video after this slide, the Astrios EQ could detect 200 nanometer beads with 80 nanometer beads near the noise floor of the system.
Running 80 and 200 nanometer beads on the Astrios EQ. This is an example of the Astrios EQ touchscreen panel as we start the 80 and 200 nanometer beads. You gently boost, and as you do, you see the 200 nanometer beads, and then you see the 80 nanometer beads within the buffer noise. We have forward scatter on the x-axis and side scatter on the y-axis. The 80 nanometer beads are above noise, but they're also within the buffer noise. As the sample is stopped, the beads are no longer visible and the instrument noise is all that remains on the touchscreen panel.
The 80 and 200 nanometer beads. Gently boost. After watching the video, it is difficult to see the difference between the 80 and 200 nanometer beads. However, this histogram shows the side scatter difference between the 80 and 100 nanometer beads. As seen on this histogram, the noise floor is visible well above the 80 and 100 nanometer beads. We have the noise floor here, and we have the 80 and the 100 nanometer beads. There are several benefits to having polystyrene beads. Uniform standardized particles, they're easily accessible and reproducible. However, there are a few drawbacks. Side scatter is dependent on the index of refraction. However, beads have a much higher index of refraction than EVs. The next calibration standard is the liposome. Liposomes can be purchased with different lipid configurations in different sizes. In the next video, the stained DII liposomes are visible on the instrument touchscreen panel. The DII liposomes, as we can hopefully see in the video, had a higher fluorescence value than
DII stained liposomes 100 nanometers on the Astrios EQ. The Astrios EQ is able to see 100 nanometer DII stained liposomes from noise on both side scatter and the fluorescence 561 channel. The instrument starts with noise visible. Then the liposome sample starts with the forward scatter and side scatter. The liposome population is visible above noise on the side scatter channel. Using the channel parameter selector, the image may be changed to the 561-579 fluorescence channel for the DII fluorescence. The stained liposomes are visible above noise on the fluorescence and side scatter channels. When the sample is stopped, the population on the fluorescence channel drops to the noise intensity levels.
Fluorescence. The DII liposomes had a higher fluorescence value than noise. Benefits to using liposomes as a calibration standard. They are biological. The index of refraction is near EVs at 1.45. They're reproducible. Additional dyes can be either packaged inside the liposome or attached to the membrane. However, there are some challenges. Liposomes do not have the mass or RNA protein content of EVs. In all, liposomes may be a good control for setting up EV experiments. A researcher could purchase pre-verified exosomes with Western blot, EM, and CD63 measurements from vendors and use those to set up their instrument and assay. Here I show MCF7 exosomes purchased from SBI to help set up the application. This particular histogram shows the noise on forward scatter versus side scatter. Here is our forward scatter channel and our side scatter channel. And what we see is there is visible noise above the threshold level, but about 10,000 events per second. The unstained EVs that you can see here are located on this particular histogram, again with the forward scatter and side scatter channels. We can see that you have the exosomes located above the noise, and we also have this population located up here. Not quite sure exactly what that population is. The stained population, however, this particular sample is stained with DII. We have the dye aggregates, we have the exosomes, and then we also either have a precipitation medium or it could also be plastic debris from the epinorth tube. So these stained EVs, as seen in this histogram, you can see the exosomes, you can see the dye aggregates, and also some sort of debris located here. Purchased exosomes are a good calibration standard, as they are biologically relevant, they match the index of refraction, mass, and density, they're effective for determining characterization and analysis settings. However, the precipitation reagent may cause interactions or confusion. For example, that particular sample, there are variation in lots, and they can be quite expensive at 50 micrograms per $350 from one of the vendors. So we've discussed three of the calibration standards, the polystyrene beads, the liposomes, and the purchased exosomes. The last one we're going to discuss is the electronic pulsar. Jim Wood from Wake Forest University and APE have developed an electronic pulsar that provides discrete trigger and signal pulses. This removes the intrinsic CV spreading from the beads and measures the background without lasers and stream. There are two adjustments, the coarse adjustment and the fine adjustment, and two different LEDs for the trigger and the pulse. In the next slide, I indicate how Alex Kaczynski and I set up the Astrios EQ with the pulsar to measure Q and B. We first ran the trigger LED into a fiber couple right here and into a pod, or precision optical device. This is where the collection happens on an Astrios EQ. The adjustable fluorescence pulse was then placed near the stream. Here is the interrogation of the Astrios. You can see the LED light located here next to the stream, and it's illuminating but not touching the stream. Different fluorescent pulse values were selected and run independently, measured by the oscilloscope. 8 peak beads were also run at the same voltage at the same time to determine the MESF values. The electronic pulsar is useful for instrument calibration for Q and B. This is an example of a histogram taken where we have the gray population as our 8 peak beads. And then we vary the output of the LED pulsar from 1 volt, which is our red, all the way down to 5 millivolts. This simulates an 8 peak bead. However, at the same time, you don't have to purchase beads, so it's less expensive. It makes it less complicated to measure Q and B because you don't have the intrinsic CV values. However, there are some challenges. If you want to determine MESF on your system, you have to make sure and run 8 peak beads, and it is not biologically relevant. So bringing these 4 calibration standards together, we have the liposomes and purchased exosomes. They are effective for application development. However, the electronic pulsar and polystyrene beads are something you can have around the lab any day, and they're definitely better for instrument standardization. They're very reproducible, easy to use types. So we've discussed all the different parts of the calibration. It is key to remember that running EVs on a flow cytometer requires clear understanding of the instrument capabilities and the instrument and the application standards that are required to validate the data. We have only discussed the analysis of EVs on a flow cytometer, but can the instrument be used to isolate EVs? Before discussing flow cytometric isolation of EVs, what are the current techniques for EV isolation? The gold standard for EV isolation is the separation of EVs via serial ultracentrifugation. And specifically noted here is the Beckman Coulter Optima XPN and the 70 Ti rotor, or others you can use. I will run through a typical protocol for human serum serial ultracentrifugation. So if we isolate EVs from human serum, just a moment. The human serum is isolated in gel clotting tubes. It's sat room temperature for 20 minutes. Then it's spun for 30 minutes at 900 G on our Beckman Allegra. And the serum is removed from the tube. You have the gel clotting tube, and you place it in a 50 ml tube. Equal amounts of PBS are added. The tubes are spun at about 12,000 G for 45 minutes to 2 hours on the Beckman Optima XPN. And then the supernatant is removed. This is where your EVs are, is in the supernatant. And then they spin again at 100,000 G for 70 minutes to 4 hours. You have a range there, but definitely going over 4 hours, you might be grabbing too much protein. The pellet that's in those tubes contains the extracellular vesicles. The ultracentrifugation EV collection competes directly with the precipitation method via a similar product as seen on this slide. This is, for example, the total exosome isolation kit. And I'll run through the same procedure that you would do with human serum. We go through the same process of collecting the serum. However, the last few steps are slightly different. You add a 200 microliter reagent for 1 ml, which is about $10 per sample, according to the web page. Then you wait for 30 minutes, and then you spin for 10 minutes at 1,000 G. And the pellet contains EVs and precipitation medium. So these are the two competing technologies. And the researchers at Van Duyn in September 2014 went through and analyzed the effectiveness of the ultracentrifugation bulk methods versus precipitation. They compared the 4 methods: ultracentrifugation, ultracentrifugation with density gradient, and the 2 precipitation methods, the total exosome isolation and ExoQuick. The UC with the OptiPrep gradient provide highest purity exosomes, noted here and here. And the UC provided the second highest yield, located here. Although more labor intensive, the UC methods both provided better isolation methods. After describing the isolation of EVs from UC or precipitation, how does the flow cytometer compare? In the following section, I'm going to describe how the flow cytometer can isolate single EVs with additional considerations to keep in mind. Users should understand the noise floor on their system for scatter and fluorescence parameters, the effects of coincidence, or when 2 EVs are seen in the laser simultaneously, and make sure to run a test polystyrene sort, and keep in mind additional controls that might be required. The noise floor might be affected by numerous particles as seen in the particles in the sample or sheath media. As seen on this histogram here, we have 2 different backgrounds. We have one that are caused by filtering and the other one that's caused by delivery methods, examples in tubes. So if you look here, if you see a lot of red, it means we have a lot of background. With the sheath only, there's small particles located on the left-hand side. Tap water, it's very heavy in particles. There's some with plastic debris. And then we have filtered into a glass tube the sample media. So as you can see, the only one that provides close or near to sheath level is the filter into a glass tube. It's also very important to pay attention to what type of delivery method you're using, either a glass syringe, a plastic pipette, a glass beaker, or even a graduated cylinder. When we are looking at the noise floor, the other thing we have to understand is we're using a sorter. A sorter requires you to oscillate the stream at different drop drive frequencies and amplitudes. For the Astreos EQ, we definitely don't suggest changing the frequency, but you may change the drop drive amplitude. This is an image that is on the touchscreen panel of the Astreos. What we did is we looked at the noise floor and how, if you increase or decrease that amplitude, how it changes the noise floor on your system. So for example, we have the red population located here. That is without any drop drive on. This would be if we were just analyzing our extracellular vesicles. However, you can see as you increase the drop drive, you also increase the noise. As the drop drive increases, you have an increase in the population, and you may actually get two populations instead of one. The drop drive is required to isolate EVs, but understanding how the components may affect EV isolation is key. Coincidence. As understanding the noise floor is integral in testing for EVs, being able to see one EV instead of two is even more essential. As a laser spot for most cytometers is between 5 and 25 microns, the possibility of multiple 200 nanometer particles going through this laser spot at the same time is high when the sample is concentrated. As seen in the next video, the coincidence or swarming is caused by EVs going through the laser at the same time. This is avoided by properly diluting the same and measuring the side scatter log height values. Some publications may require a concentration curve demonstrating that the side scatter log height values are steady with decreasing concentration, indicating an absence of coincidence.
In this video clip, I artificially created swarming to show the effect it can have on the forward scatter and side scatter of the exosomes by boosting the sample. After stopping the sample, the noise population is visible on the forward scatter and side scatter. When the lasers are shut off, the population diminishes further. The noise population is visible on the
So now that we've seen a video on swarming, it's very important that we control for that when we're working on our flow cytometer. It is also important to execute a test sort to determine if the instrument can sort small particles. As seen in this next histogram, there are 81 nanometer beads and 200 nanometers sorted on forward scatter and side scatter. The resultant sort output was then run for sort purity. The 200 nanometer beads, as you can see, are above noise. However, the noise is impacting our purity measurements. Generally, you run purity, you hide the noise, and you can't do that. So what we're going to have to do is we're going to need an additional analysis package due to the nearness of that population to the noise. These sort outputs were then analyzed by dynamic light scatter on the Delcimex. The dynamic light scatter data indicated that the 80 nanometer bead population, sort output from the Astreos EQ, had 80 nanometer beads in it, which feels good. And it also didn't have any 200 nanometer beads. Now the 200 nanometers we sorted out had 200 nanometers, as seen it was measured at 195.3, but it also had a few contaminants located on the lower left-hand side. Setting up the experiment with additional controls helps validate the EV sorting data. These include sample media with no EVs, unstained EVs, and stained with no EVs, as well as calibration and fluorescence standards. Having all of these different controls helps make sure that you're looking at EVs and not diaggregates, buffer noise, or even electronic noise. To sum up, researchers could use flow cytometric sorting for single EV isolation, but it's not effective for bulk isolation. It is selective for EVs, the purity is high, but the yield will be low. There are challenges to isolate EVs, including the sort speed dependencies on the drop drive frequency. For example, if you run a 70 micron tip at 99 kilohertz drop drive, the maximum speed is about 70,000 events per second, and of that, you have an extracellular vesicle in every other drop. Now, the other issue is that the droplet volume of the EV is large, so you may get 150 milliliters of your sample, so you might need a sample concentrator. And to do the purity, you may need to use a different method, such as dynamic light scatter. The isolation methods for extracellular vesicles. To review, the isolation of EVs is valid low yield, high specificity EV isolation method. However, ultracentrifugation with or without the density gradient provides high yield, high purity EVs. It is all well and good to isolate EVs using this method, but when I look at a tube, I can hardly see that pellet, much less understand what is inside it and its concentration. In the next section, I discuss the methods of analyzing the EV pellets and sample using current and new techniques. There are many methods to analyzing EVs after isolation. I'm going to include a few of the many methods. The first method is the BSA assay, which is a very basic method for measuring protein concentration of the sample. It's more of a heuristic representation of the isolation and doesn't give you numerical values. You don't know if that's actually blood serum protein. And so it's effective as a quantitative understanding of the measurement. Now, there also is this CD63 capture beads. You can purchase them from Invitrogen, and it confirms that you have EVs in your sample, but it is a population that can be run. Alternatively, users may use Western blot and take samples to EM. I'm not focused on those two. I would like to focus, however, on the last part of the presentation on using dynamic light scatter, which is great for measuring initial size and population, and analytical ultracentrifugation, which gives us this high resolution population analysis. This particular technique has not been used in many research publications that we've seen. So measuring the initial size and population using dynamic light scatter. This is the DelsaMax Pro. And with the dip cell that comes with it, it only requires 55 microliters of the sample to be analyzed by dynamic light scatter. It measures the intensity of the light scattered by the EVs in the sample as a function of time. The system provides the average size distribution, and polydispersity of molecules in the particles of the solution. To note, however, that the resolution capabilities of most DLS instruments is 1 to 3, such that a 100 and a 300 nanometer particle may be resolved, but populations that are closer may not. I took 2 fluorescently labeled samples. As you can see here, these are HeLa extracellular vesicles stained with PKH26. There are 2 different populations. We have the high stain population and we have the low stain population. I took both of those samples and I placed them into the dip cell for the DelsaMax. As seen on the next slide, the low population was much smaller than the high population. So this is what you would get for your DelsaMax. Here's my low sort. You see that the low population has about 137 nanometers as the size of the population. But on the other hand, the high sort has a population of 253 nanometers. I also measured the purchased exosome population, our calibration standards, MCF7, on the DelsaMax. As seen on this histogram, the MCF exosome population had variable size ranges from 122 to 246, and the smaller population is visible on the lower end. The DLS systems work well for populations with small size ranges. But giving a broader idea of the population of interest of the sample. The researcher needs to know, it's going to be 1 to 3. So you have 100 and 300 nanometers that may overlap. But it will work to give you a quick idea. Did I get anything in my tube, or is there nothing in my tube? And what sizes, what range of sizes are we seeing? So it's definitely very helpful to use dynamic light scatter for those different items. Now we could also look at extracellular vesicles using analytical ultracentrifugation. Now this is a protein chemistry technology, and it does give us high resolution. So the analytical ultracentrifugation is used for quantitative analysis of macromolecules in a solution. Here we have pictured the Beckman Coulter Proteome Lab XLAI. It's especially good for protein aggregation, antibody purities, determination, and mass. Researchers can use this for a broad range of applications to study biomacromolecules. Extracellular vesicles seems like biomacromolecules to me. This is an image of the inside of an AUC cell, where we put the reference and we put your solvent in here. And you actually measure how this particular sample compared to its solvent is sedimenting. There are 2 different methods for analytical measurement on the AUC, interference and absorbance. The interference module measures the change in index of refraction between the solvent and the sample solvent in a 2-sector sedimentation velocity experiment. Similar to the side scatter that we think of on a flow cytometer, it detects all particle populations within the sample as long as the concentration is around 0.5 micrograms per milliliter or greater. It has higher resolution than the side scatter that we see on the ASTRO-CQ. And it provides population measurements as the population sediments or sedimentation velocity, or equilibrates at different g-forces. And so now we'll hopefully transfer to another video that shows you how a BSA sample at 5 milligrams per milliliter is sedimenting at different time points. This is how we look at the absorbance module. Now, in the absorbance module, you see that it measures from 190 to 800 nanometers, and that absorbance of the population at a given radius. This is similar to your high-tech spectrophotometer. The xenon flash lamp will take up to 3 unique absorbances per cell per scan. So you can look, for example, at 210 for lipid, 260 or 280 for your DNA and RNA, and you can determine that quite well. It's also similar to a high-tech spectrophotometer that measures absorbance of sedimenting populations. As with flow cytometry instrument calibration, we tested the AUC using most of the calibration standards described previously. The only standard we didn't employ was the electronic pulsar. The remainder of the calibration standards were applied to the AUC technology to determine if the EVs could be measured. We first tested the polystyrene bead sedimentation velocity on the interference module. Before proceeding, I'd like to point out the access labels. This is an open source modeling program for AUC data called SedFit. And I am fitting the data to a sedimentation velocity value. We have the concentration located on the y-axis, and we have the S value or sedimentation coefficient found on the x-axis. The program determines the concentration of the population with each sedimentation value dependent on the slope of the interference data under the curve. So for example, here are our standard beads. We ran 22, 58, and 104 nanometer beads individually and together, as seen in this graph, to determine if the resolution of the interference module was great enough to see the 30 nanometer resolution. As seen in the sedimentation curve, there are 3 populations. We see 22 and 58 and 104, and they actually have multiple populations of each size. The goal of this calibration standard was to determine if the interference module could measure the difference in size resolution. However, polystyrene mass is much different from EVs, providing a much different sedimentation coefficient. So the nice thing about the polystyrene bead sedimentation, of course, is you can see these, but you can't sediment them the same way you would sediment EVs. We moved on to a more realistic control, manufactured liposomes from Formimax. The 100 nanometer liposomes had a much lower sedimentation coefficient, which at 4.7 versus 100, for 65% of the population. As you can see here, here's our sedimentation and the data that resulted from that. You see our sedimentation velocity, the percent of the population, the mass, and the radius that it measures. The 65%, and you can see the histogram over to the left-hand side. It gives us this pretty clear, decent size. However, what we found is that the sedimentation of the liposomes is faster than EVs, but it was much more similar than the polystyrene beads. Liposomes were also detectable not only on interference, but also absorbance because of the lipids. Finally, we tested and developed the AUC analysis by utilizing the purchased exosomes, MCF7, by running the samples at different speeds from 3,000 to 50,000 RPM. The data was then analyzed in SETFIT for concentration and sedimentation velocity to determine the best resolution, or separation of peaks. The optimum speed was 25,000 RPM, with the interference for the index refraction resolution and the absorbance at 210 nanometers, 260 to 280 nanometers for protein and nucleic acid. Multiple populations were visible with the modeled Stokes radius of 20 to 92 nanometers, and we determined that the purchased exosomes were effective for application development. As seen on the right-hand side, it gives you multiple populations which correspond to the histograms on the left-hand side. In those, you have percent populations and mass and the modeled radius values. This is radius, not diameter, so we're running, between 60 and 180 nanometers for our extracellular vesicles from the MCF7s. Finally, we went through and determined these particular exosomes and how well that they worked. Now we wanted to move on to the exosomes we sorted on the flow cytometer. So I analyzed the sorted HeLa extracellular vesicles from the Astrios EQ sort on the AUC. As we've seen in this histogram previously, I placed the low and the high sorted fractions into our AUC cell and centrifuged them at 20,000 RPM for about, I think it was about 14 hours. It could be as low as 5 hours, but up to 14 hours. We also ran some controls. As we discussed in the flow cytometry section, controls are incredibly important because these particles can be confused with all other types of particles. So we run controls such as buffer only, no EVs, and dye only. I ran several different controls to eliminate non-specific noise or aggregates. Some of the controls you can see here, I didn't put all of them up. For example, on the absorbance, we looked at the stain only. So pKh 26 only absorbs at 550. Well, there's a very, very slight response with our control. Interference, however, does see that we see particles, and that might be the buffer left over or aggregates that we see on the dyes. On the same, I also looked at unstained HeLa EVs at the 550 nanometers. We might get a slight response, but you're not seeing huge sedimentation curves that you see on the right-hand side with interference. So as outlined on this slide, it is the general protocol for analyzing EVs on the AUCs with HeLa extracellular vesicles in PBS as a solvent. As a note, if you use a different solvent, let's say alcohol, for example, your extracellular vesicles will sediment way faster than they would if you were sedimenting on, in PBS or even something thicker like maybe with 5% glycerol. We centrifuge the two-sector cell overnight at 25,000 RPM. We measure the interference at 0.5 micrograms for velocity experiments, and we also looked at different absorbance. I mean, think about spectrophotometer. You can do this as the thing is spinning round and round and round. It'll measure your absorbance of your particles on the radius of that cell. We looked at 205 to 210, depending upon lipid type, that the absorbance is different. 260 to 280 for the protein and nucleic acid. Then we also were interested in the absorbance of the two different dyes. For example, PKH 67 is 490 and PKH 26 is 550. I analyzed the data in ZFIT and overlaid the concentrations versus velocity for all the measurements and determined the concentration and groups EVs of populations. So here we have a graph. And what this is it's the sedimentation curves overlapped between the four different types. We have the HeLa interference, low sort here, interference. We have the PKH 67. We have the HeLa 210, or the 210 absorbance, and the 550 absorbance. And as you can see from this particular location, we have several different groupings. Some which have, for example, this one only has lipids. So you could think about that as maybe perhaps a lipid contamination. You have some that are only dyes. Well, that could be your dye aggregation. So it's telling you the concentration in your sample, what is extracellular vesicles, what is probably buffer, what is dye, what is lipid. So therefore you can use those to determine how your extracellular vesicle prep or method is working. The groups are separated by the diameter. You can see here on this particular table, I have the different diameters and whether or not they were located in the interference, lipid, PKH 67 or 26 location. Concentration could aid in this method development, such as things that you can think about. How could you use the AUC to help develop your EV method? You could determine, well, is there enough stain in our EVs? We'd have more absorbance. Are there lipid contaminants in your sample? How well did the ultracentrifugation versus the OptiPrep gradient with ultracentrifugation clean up your sample? Are there more contaminants with precipitation? Did I actually sort EVs or buffer debris on my flow cytometer? Do I have plastic contamination in my sample? Do the EVs aggregate? Instrument and fluorescence standards are required for effective EV analysis sorting using the AUC to look at this data and experimental controls. So we looked throughout this presentation, we've discussed three main categories: extracellular vesicles and flow cytometry, how to isolate EVs, and looking at EV characterization. On the flow cytometer, the instrument and fluorescence standards are required for effective EV analysis and sorting. Experimental controls lend validity to your data. If you're going to isolate, the ultracentrifugation with OptiPrep gradient provides purest EV isolation, but our flow cytometric sorting is great for selective EV isolation on the Astreos EQ. And finally, characterizing by doing protein quantification and captured beads measures bulk EV quantity. DelSiMax DLS provides initial population sizing information and quick analysis, and the AUC provides multi-absorbance interference, and it provides high-resolution analysis of the EV populations and dye aggregates. Before I finish, I want to make sure and acknowledge definitely the Beckman Coulter Life Sciences Research and Development Team. Also, we had some collaborations with Dr. Jennifer Jones at the National Cancer Institute at NIH, and also LabRoots for hosting this.
Thanks for that great presentation, Carly. Before we get started on the question and answer session, I would like to remind our audience that you can submit questions by typing them in the Q&A box at the lower left of the presentation window. Several good questions have already came in. Our first question is, could you sort EVs based solely on size and side scatter so they could be functionally studied later? suggesting that the antibodies might likely alter the functionality of the EVs.
Yes, so there is some, it's somewhat effective to sort out the EVs on side scatter. However, it does find that sometimes you might get some contamination with that particular particle. However, you know, we have effectively sorted out extracellular vesicles just on side scatter, not with the stain.
Great, thank you for that answer. Our next question is, in the literature, microparticles, extracellular vesicles, exosomes, and apoptotic bodies are used interchangeably to describe these vesicles. However, some papers distinguish different sizes and sources of these vesicles using specific names. Do you follow any standardized method in naming?
As we discussed in, I think, slide 3, there are, as we've seen, a huge variety of extracellular vesicles. And I focus on exosomes, which are 50 to 100 nanometers, and they are generally released via a multivesicular body out into the cytosol. Another question, Chad?
Yes, we've got one more question here. The question is, are exosomes solely defined by size, or is there additional requirements to qualify as an exosome?
Absolutely. So no, they are not defined just by size. You have to make sure that your extracellular vesicles have been confirmed by either EM, or look at the Western blot for specific antibodies such as CD63, CD9, or CD81. Those will confirm that you actually have exosomes and not just junk or debris in your sample.
Thank you very much.
Thank you, everyone, for attending our seminar today. We'll have to see if Chad has any additional questions. Thank you very much, the audience, for being here today and listening to our conversation. If you have additional questions, please put those towards Chad Schwartz. And we thank you very much for attending our seminar today.